Hello, everyone, and welcome to today's live broadcast, Sensitivity, Selectivity, and Speed, Solving Analytical Challenges in Endocrinology Using LC-MS for Clinical Research, presented by Dominic Foley, Senior Scientist at Waters Corporation. I'm Benjamin Dugas, the Senior Marketing Manager for Clinical Diagnostics at Waters Corporation, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Waters Corporation. For more information on our clinical solutions, please visit us at waters.com/clinical. At Waters, we understand that clinical diagnostics is more than collecting data, it's making a difference in someone's life. This is why we provide clinical LC-MS solutions that you can trust in every step of the workflow. Now let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. Additionally, we will be conducting a few polls throughout our presentation, and we appreciate your participation. If you have trouble seeing or hearing the presentation, click on the Support tab found on the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Following this presentation, those credits can be obtained by clicking on the tab at the top right and follow the process to obtain your credits. I'd like to now introduce our presenter, Dominic Foley at Waters Corporation. Dominic Foley is a senior scientist in our clinical scientific operations team working at Waters Mass Spectrometry Headquarters in Wilmslow, UK. Dominic is specialized in the development of LC-MS clinical research applications with a particular focus on steroid hormone analysis. Dominic has worked in separation sciences for 13 years, following his ambition of improving the quality of healthcare. For a complete biography on Dominic, please visit the Biography tab on the top of your screen. With that said, I'd like to hand it over to Dominic to start his presentation. Dominic?
Thank you, Ben. I would like to welcome everyone to this webinar today. Analytical sensitivity, selectivity, and speed are critical parameters in most analytical methods. The challenges we observe in endocrinology research can be solved by leveraging the sensitivity, selectivity, and speed of LC-MS technologies. Today, I want to provide you with an overview of how higher sensitivity tandem MS platforms can be used to meet the demands of a clinical research environment. In this presentation on sensitivity, selectivity, and speed, solving analytical challenges in endocrinology using LC-MS for clinical research. There are three main learning objectives today. The first objective is to understand the analytical benefits of using LC-MS for endocrine methods over other more traditional platforms, including immunoassay. The second objective is to show how we can incorporate selective sample preparation workflows to improve analytical performance of the method using LC-MS systems. The final objective is to demonstrate how offline automation can be leveraged to improve method performance and optimize efficiency in LC-MS labs. First of all, I wanted to cover the first learning objective by discussing the benefits of using LC-MS methods over other platforms, particularly in regards to analyzing endocrine analytes in clinical research. I will outline the Waters LC-MS IVD systems and the capabilities of these systems based on analytical sensitivity requirements in analyzing endocrine analytes. I will discuss examples of the benefits of using LC-MS/MS in endocrinology clinical research applications, which includes steroid hormones in dry blood spots, estrogens in serum, and plasma catecholamines and methanephrines. Finally, I will summarize the presentation to highlight those key learning objectives again. Now I would like to highlight the key benefits of using LC-MS/MS technology in clinical research of steroid hormones. This slide shows the steroid pathway, which contains many structurally similar steroid species. Using more traditional platforms, such as immunoassay to analyze samples, cross-reactivity of steroids can occur due to the lack of specificity of the antibodies being used. This can lead to poor analytical precision and accuracy, particularly at lower physiological concentrations. In addition, these platforms, although fast, cannot only analyze a single analyte at once, leading to increased time and cost if multiple analyte test methods are required. The beauty of using mass spectrometry and its ability to mass detect is that greater selectivity can be achieved for these steroid hormones, while also simultaneously analyzing more than one steroid hormone at once if required. As many steroid hormones are isomeric to each other, the use of tandem MS can improve selectivity even further by fragmenting the analytes into product fragments, thus providing detection through a transition or multiple reaction monitoring approach. However, tandem MS alone isn't infallible. Even with this level of selectivity, isomeric interference can still be observed as many analytes may share the same MRM transitions. Those analytes highlighted in the same colours here are examples of this. Higher resolution mass spectrometry would also suffer from the same problems due to interference from these steroid isomers. In addition, acetophenars are also detected using MS/MS, and these can also cause interference, particularly with those with analytes only 2 Daltons apart, as shown here. Therefore, to attain the levels of performance required, an additional separation technique is needed. The technique we use is liquid chromatography. For many of those analytes highlighted in the previous slides that share the same MRM transitions, we are able to separate them based on their chemical properties. In these examples here, differences in the positioning of the hydroxyl groups in the isomers allow separation of the analytes using UPLC. Therefore, it is critical to use this orthogonal technique with tandem mass spectrometry to minimize interference in steroid analysis. Before I move on to some examples of LC-MS/MS technology being applied to endocrinology research applications, I want to briefly cover the specific systems being used for LC-MS/MS analysis in this presentation. Here is the family of the Waters IVD systems for clinical research analysis. The foundation of the IVD family is the Acuity UPLC I-class Xevo TQ-D, which is a reliable, robust, and has a proven track record of performance. The mid-tier Acuity UPLC I-class Xevo TQ-S Micro provides excellent sensitivity in a compact system. And at the head of the family is the Acuity UPLC I-class Xevo TQ-XS system. This has replaced the Xevo TQ-S system in the family and is the most analytically sensitive tandem mass spectrometer available. This slide illustrates how we would relate the performance of these IVD systems to the sensitivity requirements of the classes of endocrine analytes. The Xevo TQ-D system is able to analyze a number of well-characterized analytes such as cortisol, testosterone, and 25-hydroxy vitamin D. The Xevo TQ-S microsystem is able to analyze these in addition to more challenging analytes such as metanephrines, catecholamines, and aldosterone, which require detection at lower physiological concentrations. The Xevo TQ-XS is capable of analyzing all of the analytes shown here, including estrogens, thyroglobulin, and the active metabolite 1,25-dihydroxy vitamin D. In order to meet the demands of more challenging applications, higher performance LC-MS platforms are needed to meet the requirements of speed, analytical sensitivity, and selectivity. MRM and chromatographic selectivity can be applied in the same manner across all the Acuity UPLC Xevo mass spectrometer systems. However, a high-end platform such as the Xevo TQ-S Micro and Xevo TQ-XS are necessary to avoid the trade-offs between sample throughput or speed and analytical sensitivity requirements of a method. Now I would like to cover examples of applications where we are striving for either analytical sensitivity or speed while maintaining selectivity of the methodology using LC-MS. These examples include a range of endocrine analytes in either dry blood spots, serum or plasma. Dry blood spots are an established microsampling technique providing a low-cost approach of collecting, shipping and analysing samples for clinical research. Immunoassays are used as a primary testing methodology for DBS samples in steroid hormone analysis. Although rapid, they sometimes require follow-up for confirmation purposes. The focus for an LC-MS method for steroid analysis in dry blood spots will be a high throughput method, similar to immunoassay, but with the ability to measure more steroid hormones in a selective manner. Faster analysis requires greater selectivity in sample preparation, with automation of the extraction providing a robust and reproducible sample preparation workflow. The analytical sensitivity of the UPLC I-class and Xevo TQ-S microsystem will enable analysis down to relevant steroid hormone concentrations using 3 mm blood spot punches. The primary goal of this method was to examine 17-hydroxyprogesterone, the 17-OHP, androstenedione, cortisol, 11-deoxycortisol and 21-deoxycortisol, which are impacted by the 21 and 11 hydroxylase enzymes in the steroid pathway. A reduction in 21 hydroxylase enzyme leads to elevated 17-OHP, 21-deoxycortisol and androstenedione with reduced cortisol. A reduction in 11 hydroxylase leads to similar changes to 21 hydroxylase deficiency, but 11-deoxycortisol is elevated as opposed to 21-deoxycortisol. Examination of multiple steroid hormone concentrations improves our understanding of these pathways. These steroids are also non-polar, making selective sample preparation strategies for optimum analytical sensitivity challenging. Therefore, introducing a method to remove matrix effects is a route forward, in this case using mixed mode solid phase extraction, or SPE. Oasis mixed mode anion exchange SPE was used for this analysis. Previous steroid hormone applications have used this approach, and it was also adopted in this instance. Ion exchange occurs at high pH, trapping acidic interferences, which have been previously shown to contribute to ion suppression and background noise on the system. The steroid hormones are retained by reversed phase interactions, and these are eluted with increased organic solvent, which leaves the interference trapped on the SPE chemistry. This is a summary of the methodology we adopted to meet the challenge of analytical speed and selectivity, but achieving a necessary analytical sensitivity too. Sample preparation was automated on the TCAN liquid handler, with 3 mm blood spots being mixed in internal standard solution and diluted. SPE was performed using the Oasis MAX SPE plates, with the sample loaded and washed, and steroid hormones eluted, diluted, and directly injected onto the system. Separation was performed using the Acuity UPLC I-class with its Cortex C18 2.7 micron 2.1 mm by 50 mm column with a Vanguard pre-column. Mobile phases were ammonium fluoride in water and methanol, with a flow rate of 1 ml per minute, providing a rapid separation time and sample throughput. Tandem mass spectrometry was performed on the Xevo TQ-S micro system with MassLinks version 4.2. Utilizing all the technologies available to us, automation of this SPE protocol was performed in less than 90 minutes per plate, allowing for extraction of 4 plates in a typical working day. A rapid 1.3 minute LCMS method with a cycle time of less than 2.3 minutes was created with the separation of the relevant isobaric steroids, including 11 deoxycortisol, corticosterone, and 21 deoxycortisol. This allows for the analysis of 384 samples in less than 15 hours using LCMS-MS. This slide shows the typical workflow you might expect during a typical day of analysis of 4 full plates of DBS samples. The entire workflow from start to finish for a single plate is less than 6 hours. And for 4 96-well plates, this can be prepared and analysed within 18 hours. Proof of concept experiments were performed for this method that included linearity, analytical sensitivity and precision. Calibration of androstenedione and 11-deoxycortisol was performed from 0.5 to 500 nanograms per ml, and for 17-OHP cortisol and 21-deoxycortisol from 1 to 500 nanograms per ml. LOQs, also shown in the chromatogram here, are based on signal to noise of peak to peak, where 0.5 nanograms per ml for androstenedione and 11-deoxycortisol and 1 nanogram per ml for 17-OHP cortisol and 21-deoxycortisol. In-house DBS samples were also prepared at 2, 5, 50 and 400 nanograms per ml, with total precision and repeatability evaluated in 5 replicates over 5 occasions. Total precision and repeatability for the 5 steroid hormones was less than 9.3%, demonstrating excellent reproducibility of the method across the concentration ranges. So in summary, we believe we have met the analytical challenge associated with this type of method. We obtain excellent analytical sensitivity in DBS, down to 0.5 nanograms per mil for the steroid hormones, and selective offline automated sample preparation can improve lab workflows and removes matrix interference, providing greater sensitivity of the final extract. Rapid separation with a runtime of less than 2.3 minutes cycle time enables analysis of 4 plates in less than 15 hours, making this method an ideal candidate for rapidly evaluating steroid hormones in dry blood spots for clinical research. Next, I would like to cover the analytical challenges associated with the analysis of estrogens in serum. The focus for this method is analytical sensitivity, where guidelines have been issued that methods require reproducible measurement down to 1 picogram per mil or 3 picomoles per litre. In order to meet this challenge, selective sample preparation is required, using LLE or liquid-liquid extraction, or SPE, and the top-performing Acuity UPLC I-class and Xevo TQ-XS system for best-in-class analytical sensitivity. Mobile phase modifiers can improve analytical sensitivity further. Ammonium fluoride additives in the mobile phase have been shown to provide significant improvements in analytical sensitivity for this method. The estrogens themselves are generated from the androgens androstenedione and testosterone through the action of aromatase. The efficacy of aromatase inhibitors can be studied in clinical research by examining the changes in estrogen concentrations through aromatase inhibitor action on the pathway. Analysis of estrogens at such low concentrations can be problematic for less selective platforms, such as immunoassay. Use of LC-MS can help overcome such a challenge. Much like the other steroids previously discussed, the estrogens are non-polar, making, again, sample preparation a challenge for optimum analytical sensitivity. In this instance, with analytical sensitivity the main challenge for the method, analytical alternative sample preparation approaches were evaluated, which included SPE and LLE, with LLE using a combination of different extraction solvents for evaluation purposes. Typically for LC-MS methods, optimum analytical sensitivity is achieved using more selective techniques such as SPE and LLE. And in this instance, both techniques were evaluated, and LLE with hexane ethyl acetate was found to provide the highest analytical sensitivity when considering both estradiol and estrone together. The addition of ammonium fluoride as an additive makes a significant difference in analytical sensitivity for the estrogens. Compared to mobile phase with no additives, shown in red, there is a greater than 5-fold increase in analytical sensitivity in electrospray ionization negative ion mode for both estradiol and estrone. So this is a summary of the methodology we developed to meet the main challenge of analytical sensitivity with selectivity. Important aspects of the method are highlighted in blue. Sample preparation was performed by adding internal standards to 250 microliters of serum. LLE with hexane ethyl acetate mixture was added to the samples and shaken for 5 minutes, with the estrogens partitioning into the organic phase. Samples were centrifuged and then 700 microlitres of supernatant was transferred to a 96 well plate with glass vial inserts. This was then evaporated and reconstituted. The glass vial inserts, in this instance, reduced adsorption during evaporation compared to polypropylene 96 well plates. Separation was performed using the Acuity UPLC I-class with a Cortex phenol, 2.7 micron, 2.1 mm by 50 mm ID column. Mobile phases were ammonium fluoride in water and methanol, with a flow rate of 0.3 mL per minute, providing a separation of estradiol and estrone. Tandem mass spectrometry was performed on the Xevo TQ-XS system, providing us with the best possible analytical sensitivity for this analysis. The key performance characteristic is shown here, which is analytical sensitivity. The limit of quantification was 3 picogram per mil, or 10 picomoles per litre, 11 picomoles per litre for estradiol, which showed a signal to noise of peak to peak greater than 10 and a CV of less than 20%. The limit of detection was 1 picogram per mil, with a CV of less than 20%, but only a signal to noise greater than 5, but less than 10 to 1. The chromatogram here illustrates an overview of this testing, which demonstrates we are able to differentiate between a blank serum sample and a sample at 1 picogram per mil. For other performance characteristics, in-house serum QCs were prepared at 10, 75 and 750 picograms per mil, with total precision and repeatability evaluated in 5 replicates over 5 occasions. Total precision and repeatability for the estrogens was less than 4.8%, demonstrating excellent reproducibility of the method. Comparisons were also performed against samples from the CDC Hormone Standardization Program and NEQUAS scheme for estradiol. Deming regression and Altman blend analysis demonstrated excellent agreement with a minimal bias within 4% for both sets of comparisons, with only a statistically significant proportional bias noted for estradiol when compared to the CDC Hormone Standardization Program concentrations. Certified reference material was also evaluated in triplicate over 3 concentrations, and these returned results within 4.9% of the nominal concentrations provided. So in summary, for this particular method, using a combination of selective liquid-liquid extraction sample preparation, ammonium fluoride in the mobile phase, and the Xevo TQ-XS system, we were able to detect 1 picogram per mil concentrations for both estradiol and estrone. Precision assessments demonstrated reproducibility of the method across the concentration range, and excellent agreement was noted for external quality assessment schemes. And the final method I would like to cover today are the analytical challenges associated with the analysis of plasma metanephrines and catecholamines. As with the estrogens, the focus for this method is analytical sensitivity, where these compounds exist at low physiological concentrations. The polarity and stability of these analytes make sample preparation a challenge. A selective extraction is required that can remove these ionic polar analytes from matrix at lower pH conditions, due to their instability at higher pH. Chromatography also becomes problematic due to this polarity, and an analytical sensitive system is required to detect the lower concentrations. So in this case, we use the Xevo TQ-S mass spectrometry for this analysis. The pathways here show how these catecholamines and methanephrines are generated in the adrenal gland. The methanephrines are generated from the catecholamines through the action of catechol or methyltransferase. Note that these are structurally similar species, which means that typical analytical methodologies for this analysis, such as HPLC with electrochemical detection or immunoassay, can suffer from interference. As discussed previously, the use of LC-MS can overcome these types of problems. This picture also shows the log P, which, as you may know, is a measure of the hydrophobicity of the molecule. Here we are dealing with molecules that have negative log P values, which indicates the molecules are very polar and means the molecules elute early in the typical reverse phase chromatography conditions, with issues around peak shape and matrix effects. In addition, the nature of these analytes makes sample preparation more challenging for analytically sensitive analysis. We meet the challenge of sample preparation through the use of Oasis mixed mode weak cation exchange SPE. Analytes are retained by a mixture of reverse phase and ion exchange retention, but the ion exchange mechanism is a requirement for improved recovery and extract cleanliness. The ion exchange retention mechanism of this SPE plate occurs at higher than pH 5. These analytes are ionized at lower pH, typically below 8, meaning that the region of ion exchange using this chemistry is between pHs 5 and 8. Therefore, the pH change for maximum ion exchange needs to be tightly controlled using a buffer diluent. 100% organic wash can be performed to remove non-ionic species from the plate, and the analytes can be eluted in a low pH organic elution, which deactivates the ion exchange mechanism on the plate by deionizing the chemistry on the sorbent. The use of this chemistry also helps provide us avoid the use of higher pH conditions that can cause analyte instability. In relation to the challenges with the chromatography, many chromatographic methods for these analytes involve the use of hydrophilic interaction liquid chromatography separations, otherwise known as HILIC, which can be challenging to implement. Maintaining the pH of the mobile phase and SPE elution solvent is critical for reproducible retention times. In addition, the chromatography is very sensitive to water content of the extracted sample, which is illustrated in the chromatograms here, with fronting and poor baseline resolution as a result of residual water in the injection solvent. Longer equilibration times are also needed to replenish the water layer on which the separation relies upon. This doesn't necessarily fit into most laboratory workflows where reverse phase methods are typically used, which is why a reverse phase method will be preferable for the workflow, ease of use, and improved robustness. So this slide shows an outline of the methodology applied for this analysis. Again, some of the critical points are highlighted in blue. Sample preparation was performed using 250 microliters of plasma diluted with internal standard in ammonium acetate buffer. Samples were mixed and loaded directly onto an Oasis WCX microelution plate and washed prior to elution. The use of microelution technology helps minimize the volume in the elution step, thus minimizing the time required for evaporation. Samples were reconstituted prior to injection. UPLC was performed using the Acuity UPLC I-Class FTN system with a HSS PFP column for retention and separation of the analytes using reverse phase chromatography conditions with mobile phases of formic acid, water and acetonitrile. Detection was performed using the Xevo TQ-S mass spectrometer. And now I'd like to show some of the performance characteristics associated with this method. We were able to develop a fast and selective chromatographic separation using a PFP stationary phase, which fits into the reverse phase workflow of most laboratories, with all analytes separated within 2 minutes. Potential interferences such as metformin, midodrine and L-DOPA were all well separated. LLOQ experiments demonstrated analytical sensitivity limits between 1 and 10 picograms per mil for the analytes of interest, making quantification of physiological levels possible. The method was also shown to be linear for the analytes up to a concentration of 20,000 picograms per mil. We also looked at precision performance, which was evaluated through the assessment of in-house QC samples prepared at 15, 30, 750 and 1500 picograms per mil in plasma. Inter and intra-batch precision was examined over 6 replicates per occasion over 3 occasions, with a CV of less than 15.9% for all analytes across the concentration ranges, with many of these under 10% CV. So in summary, for this particular method, a reverse phase chromatographic method was employed to provide a rapid, robust separation with baseline resolution of interferences. An efficient sample preparation method using solid phase extraction provides extraction of polar analytes from plasma at lower pH in a 96 well plate microelution format, with the option to automate the method using a liquid handling system in the future. The Acuity UPLC I-Class Xevo TQ-S microsystem enables detection at low physiological concentrations of catecholamines and metanephrines for clinical research, providing better detection limits compared to entry-level systems. So, in conclusion for this presentation, use of LC-MS systems provides significant selectivity advantages over more traditional techniques used in clinical research. Use of higher-end platforms such as the TQ-S micro and particularly the Xevo TQ-XS enables users to meet the analytical sensitivity, precision, accuracy and speed requirements for the methods. Selective sample preparation and chromatographic separation helps users meet the unique method challenges such as removal of interferences, polar retention and analytical sensitivity, while leveraging automation of sample preparation, particularly in high throughput environments, which can improve lab efficiency and minimize operator error. And so, before I finish this presentation today, I would like to thank my colleagues, Heather Brown, Robert Wardle, Chen Li and John Danisal, who have all been involved in generating the methods found in this presentation. I've also attached links relating to collateral for the methods and also the Waters clinical landing page at www.waters.com/clinical, where you can find further information on the Waters Clinical Laboratory LC-MS workflow, ranging from sample handling to leasing. And so I'd just like to thank you for your attention today during this webinar, and I would like to now open the floor to questions. Thank you.
Thank you, Dominic, for this very informative presentation. So now we'll start the live Q&A portion of this webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. We have time for a few. So let's get started. First question that came in, Dominic, is have you looked into the impact of the hematocrit effect on the steroids dry blood spot method?
Thank you, Ben. So we are aware of the impact of the hematocrit effect in regards to dry blood spots, but this, in regards to the steroid methods, was not investigated. We know that dry blood spots is widely adopted, so the focus was on that particular matrix. And we are aware that other sample or micro-sampling techniques that could help negate this effect could well be adopted within this methodology too. So they include VAMs, or fixed volume blood spots from capillary collection type devices, could be used, but we didn't go into that level of detail in terms of the hematocrit effect in this instance.
Great, thank you, Dominic. So another question that came in is, have you examined any micro or nanoflow techniques for this method, for these methods?
Right, okay. No, not particularly. We haven't really looked at those techniques, as our goal was to use a standard flow system, as these are more widely adopted in clinical research labs. So the aim was to use a fairly standard UPLC setup. So any of the methods we do create will fit within the workflows of a typical lab. So in this instance, no, we haven't looked at those techniques.
Okay, great, thanks. And for the sake of time, I'll take one more question. Can the dry blood spot method be transferred to plasma or serum-based methods for steroid analysis?
So in answer to that question, yes. Theoretically, the main aspects of the method can be transferred, but when doing that sort of transfer, protein binding, particularly with the steroids, can be an issue, so that has to be taken into consideration. Robustness and matrix effects have to be considered, which means that any minor changes to sample preparation and chromatographic runtime might be required to mitigate the impact of those changes.
Okay, great. Thank you. Okay, well then I'd like to thank the audience for joining us today and for your interesting questions. The questions we did not have time for today, we will, and those who are submitted during the on-demand period, we will address these by the speaker via the contact information you provided at the time of registration. We would like to thank Dominic Foley for his time today and his important research. We'd also like to thank LabRoots in association with us here at Waters Corporation for underwriting today's educational webcast. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. At the close of the webinar, we want to direct everyone to the next Waters webinar that's on June 25th at 11 a.m. Eastern Time, entitled Mass Spectrometry, Past and Present, Emerging Technologies and Strategies for Quality Management in Today's Clinical Laboratory. So we hope to see you there, and thank you again, everyone.